Dear friends, today we are going to see about gene therapy. I am Dr. Lena, Assistant Professor, Department of Zoology, Sri Sharada College for Women, Salem. What is genes? Genes are contained in a chromosome which are in the cell nucleus. Inside the nucleus, there are chromosomes. Inside the chromosome, there will be genes. A single chromosome may contain thousands and thousands of genes. Gene is responsible for the physical and functional unit of heredity. Genes are made up of DNA. So, heredity. How we inherited from our parents? Inheritance is the process by which genetic information is carried on from parent to offsprings. That's why we members of same family tend to have similar characteristics. Parents pass on traits or characteristics such as eye color and blood type to their children through genes only. Genetic disorder. Coming to genetic disorder, it is nothing but a disease which is caused in a DNA sequence which is away from the normal sequence. It may be caused due to mutation in a single gene or a multiple gene. So if it is caused in single gene, it is a monogenic disorder, and when it is caused in multiple gene, it is a multifactorial inheritance disorder. The reason may be combination of different gene mutation, environmental factors, damage to the chromosome, maybe difference in a structure of chromosome, or changes in number of genes in the chromosome, or the genetic disorders. What are the causes of genetic disorder? It may be genetic problem which may be inherited from mother to offsprings. It may be a chromosomal problem or some infections acquired during the lifespan, or exposure to medications, chemicals, or other agents during pregnancy. Coming to gene therapy is a novel treatment method which utilizes genes or short sequences as a therapeutic molecules. Instead of using conventional drugs, we can use gene for curing the defective genes in the body. This technique is used to treat those defective genes which contribute to disease development. Gene therapy involves the introduction of foreign genes into an organism to treat hereditary or acquired genetic defects. In gene therapy, therapeutic protein is packed in a vector which transport DNA into body of a person. Simply, gene therapy is modifying the expression or repairing the damage of abnormal genes. Gene therapy and genetic disorder. So, gene therapy works by replacing or inactivating disease-causing genes. In some cases, gene therapy introduces new genes into the body to treat a specific disease. Simply, it delivers a healthy copy of genes to the cells inside the body. How gene therapy works? Gene therapy is designed to introduce genetic material into cells to compensate for abnormal genes. It may be able to introduce a normal copy of gene to restore the function of protein. A gene that is inserted directly into a cells usually does not function. So, it needs a vector. Vector is a genetic engineered which is helping in delivering of gene. It is a vehicle. Vector is a vehicle. Certain viruses are often used as a vector. Vectors can be injected to the tissue intravenously. So this picture shows the healthy gene which is transmitted inside the vector, and this particular vector is containing DNA sequence now, which is healthy gene, and it is transmitted inside the person body. What are the types of gene therapy? Types of gene therapy, there are several approaches for correcting the faulty genes in the body. We can use two different types, namely somatic gene therapy, germline gene therapy. Coming to somatic gene therapy, the somatic cells of a patient are targeted for gene transfer. In the case of effects caused by foreign gene, it is restricted to the individual's patients only. Germline therapy, the function genes are integrated into the genomes, that is germ cells. So we can divide our body cells into two types, as you all know, somatic cells and germ cells. Somatic cells, it is present in all other organs, and germ cells are present in the reproductive organs. So here germ cells, it is injected in the germ cells, that may be a sperm or egg. The targeting cells are germ cells in the case of germline gene therapy. What are the methods of gene therapy? Transfer of genes into the patient's cells. It may be outside or inside the body. So when it is outside, it is called as ex vivo gene therapy, and when it is inside, it is called in vivo gene therapy. There are different techniques. Gene augmentation therapy, a procedure for correcting metabolic deficiency caused by missing or defective gene by having a healthy gene produce the necessary product without actually substituting that gene for the absence of gene. In gene inhibition therapy, it is suitable for treatment of infectious disease such as cancer and other inherited disease. The aim is to introduce the product either in the inhibition of expression of another gene, or it interfere with the activity of the product of another gene. What are the uses of this particular gene therapy? Gene therapy helps in replacing the faulty genes and adds a new gene in an attempt to cure disease or improve your body's ability to fight disease. So, no need of taking medicine, no need of taking any other lifelong medicines. So, instead we can go for a different approach, that is, we can rectify a problem in a gene by using another supergene, that is called as gene therapy. This gene therapy holds promise for treating a wide range of diseases such as cancer, cystic fibrosis, heart disease, diabetes, hemophilia and AIDS. So this is the brief introduction about gene therapy. Thank you for listening.